investigating the effects of T consciousness fields on growth and metabolic activity of Escherichia coli by respiration activity monitoring system based on Taheri's theory. The outline of my talk is as follows: T consciousness fields, background, the aim of this study, material and methods, results, conclusion, references, and research team. In the 1980s, Muhammad Ali Taheri introduced T consciousness as a fundamental element of the universe and the origin of information, matter, and energy. In this approach, there are various T consciousness fields with non-physical entities that can be examined through laboratory experiments. The bacterial growth progresses in 4 phases, including the initial phase of slow growth or lag, exponential or log phase characterizing with maximum growth rate, stationary phase where the number of organisms remains almost constant, and the period of decline or death phase. The respiration activity monitoring system has been developed for online measurements of the respiration activity parameters such as oxygen transfer rate, carbon dioxide transfer rate, and respiratory quotient in shaking flasks. This allows us to quantify the physiological state and gain relevant information about metabolic activity of a microorganism. While entropy is commonly associated with the measurement of disorder, the observed biological order over time may be interpreted as a violation of the second law of thermodynamics. On the other hand, this physical property in living systems has always been a subject of study, particularly when discussing the role of information processes in some basic biological phenomena like growth. The Shannon entropy as the fundamental quantitative notion in classical information theory has been frequently used in biology. The aim of this study was to assess the effects of T consciousness fields on the growth phases of E. coli utilizing the respiration activity monitoring system. Moreover, the calculation of entropy allows us to understand the effects of T consciousness fields on this physical property at the level of bacterial population, and it can provide better insight into the process of death and life of samples under the influence of T consciousness fields. This experiment was carried out using double-blind method according to Cosmo Intel Research Center. E. coli was cultivated in shaken flasks with a frequency of 200 rpm. There were 4 Erlenmeyer flasks, including 2 controls without T consciousness fields treatment and 2 T consciousness fields treated samples. These flasks were screened in parallel every 30 minutes for 24 hours, and data were obtained at 46 time points. To explore the relationship between entropy and the data on the oxygen transfer rate of this bacterial strain under study, the values were first grouped into categories based on their minimum range. This grouping facilitated comparison among samples within each range, avoiding single-member distributions and allowing for inclusion of values necessary for rate calculation. Next, the probability associated with each data range was obtained by using the frequency of numbers in each interval and the total number of values in the growth phases of each sample. Subsequently, the entropy value was computed using Shannon's entropy formula for each interval on the growth chart. Although oxygen transfer rate of the T consciousness fields treated samples and controls exhibited similar behavior in the lag and death phases, there were different trends in the log phase. Carbon dioxide transfer rates of both treated samples were significantly lower than control 2, suggesting reduced bacterial metabolic activity under the influence of T consciousness fields. At the beginning of the period, the value of respiratory quotient for control was around 1.3. Subsequently, there was a general declining trend, reaching just under 1. In contrast, treated samples exhibited the opposite behavior. After remaining steady at a value of 1 for an extended period, they showed a gradual increase in the middle of the growth timescale, indicating improved survival during the bacterial growth cycle. The comparison between the oxygen transfer rates averages revealed that the growth rate of T consciousness fields treated samples was about 44% lower than controls. Moreover, the doubling time of samples under the influence of T consciousness fields increased by about 75% compared to the controls. Under T consciousness fields treatment, the entropy was on average 16.2% lower for the log phase and 2.7% lower for the death phase. In conclusion, this study revealed that samples under T consciousness fields treatment had lower respiratory quotient, longer doubling time, and decreased growth rate. In addition, these fields reduced the entropy of the log and death phases of E. coli growth. Our research team, and thanks for your attention.